Hello, everybody. Welcome back for the afternoon session. It is my very great pleasure to introduce our third keynote speaker for today, Allan Jacobson. Allan earned his PhD at Brandeis in David Gillespie's lab, where he was studying gene regulation in E. coli. And then he moved to Harvey Lodish's lab at MIT and began to study gene regulation in the slime mold Dictyostelium. This was an old-fashioned short postdoc, and shortly after, he started his own lab at UMass, where he has been for a long time. Allan is well known to most of you in this community for being at the forefront of research, both in translational control and in translation-associated messenger RNA decay pathways for a very long time. Allan's lab in the early years became interested in the role of the poly-A tail, first in regulating messenger RNA stability and then in regulating translation initiation. And they made many seminal contributions to our understanding of how the poly-A tail and the poly-A binding protein regulate these processes. Somewhere along the way, he shifted from focusing on Dictyostelium to working in Saccharomyces cerevisiae, and for much of his work, he's exploited the incredible genetic power of budding yeast to discover the core components of the nonsense-mediated decay pathway and to elucidate how they regulate post-transcriptionally mRNA levels in response to translation termination defects. His lab has also taught us a great deal about the kinds of mRNAs that can be recognized by this pathway. And this has also had very important implications for human health, where a large fraction of human diseases are caused by premature termination codons. For his pioneering and long-standing important contributions to the understanding of nonsense-mediated decay, Allan was recognized in 2023 with the Gruber Prize in Genetics, as well as many other prestigious accolades. I want to add that in addition to his seminal contributions intellectually to the field, Allan is also a pillar of our community. He's a wonderful colleague who is well known for being generous and supportive of younger scientists. And so I don't want to take any more of Allan's time, just to say welcome and thank you.
Thank you, Wendy. You're going to make me cry. So just by way of a thank you, I've been coming to Cold Spring Harbor meetings for more than half a century. In other words, since before most of you were born. This place is my favorite place for meetings, and this meeting, which I think I've attended all of them, this is my favorite meeting. So thanks to Rachel, to Daniel, and to Wendy for inviting me and for putting together such a terrific meeting. All right, so this is the disease and therapeutic session, and we'll get to that in a minute. But as the saying goes, first a little roo-roo. We're gonna talk about NMD. Okay. So in the previous century, in 1979, Francois Lecroute and Regine Lasson discovered for the first time in eukaryotes that a premature termination codon leads to destabilization of mRNA. They did it the hard way, and when we worked out methods for measuring decay rates of mRNAs in yeast, we did it a little easier. So here, with a temperature-sensitive RNA polymerase, we showed off transcription at T0. And here are two versions of the yeast PGK1 mRNA, the wild type, and one with a single base change that led to the inclusion of a premature termination codon. And so here's the phenomenon. In yeast, which is most of what I'm going to talk about today, NMD gives you somewhere on the order of a 5 to 10-fold destabilization of an mRNA. Now, Lecroute's lab did this in the late 70s. It's important to note that in labs studying hemoglobinopathies, and just a few years after Lecroute, it was generally seen that patients with hemoglobinopathies caused by nonsense mutations, they couldn't recover substantial amounts of alpha-globin mRNA or beta-globin mRNA, and surmised that something might be happening either to processing of the RNA or turnover. And Lynn McQuat, working in Jeff Ross's lab, showed indeed that one of the globin mRNAs, beta-globin mRNAs derived from a nonsense-containing transcript, was unstable. So there was this general understanding, starting with the recruit, that mRNAs could be destabilized by PTCs. But what was that all about? And the assumption for most of the 80s was that when you have a premature terminator, ribosomes fall off, and that you expose a large segment of the coding region of an mRNA, and that the cytoplasm was supposedly full of endonucleases, which then went to work, and that was what the problem was. So this changed when I met Michael Culbertson at a meeting, and I learned about his UPF mutants, which were things he was studying to understand frame shifting. And I encouraged him to think otherwise about possible destabilization of these mRNAs. And his student, Peter Leeds, came to my lab to measure decay rates in strains that had deletion of the UPF1 gene. So UPF originally was up frame shift. That's what that was all about. And so working with Stu Peltz in my lab, Peter and Stu showed that wild-type His4 mRNA was stable either in a wild-type cell or in a UPF1 deletion, but a nonsense-containing version of His4 was unstable in wild-type cells and stabilized in a UPF1 deletion, and likewise in other nonsense-containing transcript, also decayed rapidly in wild-type cells and slowly in UPF1 delete cells. We then began to pursue UPF1 earnestly, starting to do the same sort of experiment with numerous probes for different genes. And Hu Feng in my lab showed that intron-containing RNAs that bypassed the nuclear retention that got into the cytoplasm were also substrates for this UPF1-mediated decay. And so here is the CYH2 mRNA, which in wild-type cells barely manifests its pre-mRNA, but in UPF1 deletion cells, you see it. And if you just probe for the intron, it's even easier to see. RP51B mRNA, harder to see, but the same phenomenon. And MER2 mRNA, intron, again, stabilized in UPF deletion cells. So we began to think that it's not just mRNAs. Concurrently, we used the two-hybrid screening method to use UPF1 to find UPF2 and UPF2 to find UPF3. At the same time, Phil Anderson's lab was finding the SMUG genes in worms, which were analogous to the UPF family of proteins. Genetically, we showed that UPF1 is the principal regulator of the pathway. It could compensate for loss of UPF2 or 3, but not vice versa. And biochemically, Elena Conte's lab showed the same sort of thing. The activity of the helicase activity of UPF1 depended on activation by UPF2 and UPF3. Okay. UPF1, when tagged here, sediments in the polysome, largely in the polysome region of a gradient. Here it is on a centromeric plasmid, and these are all high copy. Okay? Note that if you delete UPF2, that the UPF1 sediments faster, and we can get to that later. Okay. UPF1 interacts with ribosomes, and we could, using the two-hybrid system again, show that UPF1 interacts with RPS26A, and that interaction is wiped out by some mutations in UPF1. So using ribosome profiling, what we were seeing using selective ribosome profiling, where we pulled down tagged UPF1 and the ribosomes associated with it, we saw two early phases in UPF1 association. One was a stochastic on-off of UPF1 with ribosomes, largely surveying for premature termination codons. And then a next step in which UPF2 and UPF3 join when a premature termination site is seen. Okay. If you delete UPF1 or 2 or 3, you have the same consequences for cellular mRNA levels. And they are, you get a huge stabilization of about 900 mRNAs in all of these, and very few downs. Okay? And UPF1, UPF2, and UPF3 all have the same, largely the same complement of ups, if you will. And the downs are negligible. And so this is consistent with UPF1, 2, and 3 all controlling the same events. Okay. Those 900 substrates can be broken down into what we call traditional and probabilistic substrates. So traditional, of course, transcript, which is derived from a gene having a nonsense mutation, or a transcript that has an unspliced intron, many, but not all, of the upstream ORF-containing RNAs, mRNAs with program frame shifting. Yeast even has an occasional pseudogene and a couple of polycystronic transcripts. Okay? And then the probabilistic substrates are those where a nonsense codon is encountered in the course of gene expression. So leaky scanning, bypass the normal AUG, the ribosome now initiates out of frame, bingo, there's a premature terminator. Alternate transcription start sites, this is analogous to what Nick Gaida showed you the other day with IRFs. Okay? And elongation errors, NOCs here are non-optimal codons, and an elongation error here is a frame shift, and now you get a premature termination. So we think of NMD as a probabilistic quality control mechanism for protein synthesis. If you've got a problem mRNA, you want to get rid of it. Okay. Now, there's a rumor in mammalian cells that there's a pioneer round of translation. We don't see that in yeast, and here's one way that we demonstrated that. This is now from our older microarray data, where we created a GAL-regulated UPF2, and we start with deletion of UPF2, so all of these mRNAs that are substrates, these are just substrates of the pathway, all of these are now abundant and stabilized, and now we turn on, we add galactose at 10 minutes, 20 minutes, 40 minutes, and 60 minutes, and what you see is that all these steady state, pre-existing mRNAs, way past anything that might be called the pioneer round, decay. With Matt Sachs's help several years ago, we did the same experiment with a single mRNA. The CPA1 mRNA has an upstream ORF where in the presence of arginine, the upstream, the nascent peptide blocks the ability of the UORF to get further than the premature termination codon. In other words, it stalls it at the termination codon, and by so doing, it turns this transcript into an NMD substrate. And so here is the addition of arginine, again, in steady state, and if you add water or arginine, you can see that the arginine destabilizes this pre-existing transcript. So no pioneer round, no immune escape for NMD in yeast. Okay. Now, NMD regulates decapping of transcripts. And we learned that by doing cap IPs, immunoprecipitation of transcripts by virtue of grabbing their caps. And so here in a wild-type cell, this is input, the IP input, soup, and pellet. So most of the transcripts in wild-type cells are pelleted. That means they're capped. Okay? And same result in UPF1 cells. NMD2 is our earlier name for UPF2. Same result there and there. Okay? But now you see if you block exonucleolytic digestion, then about half the RNA is now uncapped. Not a surprise. And if you block decapping, it's all capped. And if you combine the two, it's still all capped. All right? And so now if you examine what happens if you combine all the UPFs with XRN1, you see that you now have a sizable fraction that's capped, but not exclusively. So the appearance from these experiments is that all three of the UPFs regulate decapping. Now, the way that thought became firmer was by experiments that Hu Feng did in the lab. And here, over years, he defined the binding sites on the catalytic component of the decapping enzyme, DCP2, the binding sites for UPF1. He talked about this on Tuesday. So there are two almost identical binding sites for UPF1 on DCP2, and binding sites as well for EDC3 and other decapping activators. But let's just focus on UPF1. If you delete either of the UPF1 binding sites on DCP2, you do not lose interaction in a two-hybrid screen. However, if you delete both of them, you lose all interaction. And so these are redundant binding sites for UPF1 on DCP2. Okay. And just to remind you what Feng showed on Tuesday, there is a distinct subset of the decapping enzyme that targets NMD substrates, and it is the form that has UPF1 bound to DCP2. Now, the question is, I just told you that UPF1 bound to UPF2. I also told you that UPF1 bound to DCP2. And are those concurrent, or can they overlap? So here's what a two-hybrid reaction looks like if you just look at UPF2, UPF1, nice blue spot. But in the lower panel, what we did was look at a bridged two-hybrid. So UPF1 doesn't interact with DCP1. It interacts with DCP2, but DCP2 interacts with DCP1. And so DCP1 interacts with DCP2, and you get what looks like an interaction with UPF1 because that's bridged. Okay? If UPF2 was interacting with UPF1 at that time, you would see a bridged interaction there as well, and you don't. So UPF1 DCP2 and UPF1 UPF2 interactions are distinct. And if you remember, I showed you that UPF1 bound first, and then UPF1, 2, and 3. Okay? And then what happens subsequently is the loss of UPF2 and 3 before DCP2 binding occurs. And just a little peek at what this looks like with AlphaFold's help. This is a piece of UPF2 that binds to the UPF1 CH domain. Here's that same CH domain with the two DCP2 UPF1 binding sites. And so roughly the same piece of the CH domain has to interact with these two other proteins. And so you can't do both of those at the same time. And so that's consistent with the two hybrid stuff I just showed you. Yeah. Now what happens if the UPFs are gone or inactive? All right, I showed you the substrate data that you accumulate a lot of substrates, but are there translational phenomena? Yes. So here we're doing selective ribosome profiling, so we're overexpressing UPF1 or mutants of in the NMB pathway. Then we're IPing ribosomes, and then we're asking what codons are those ribosomes parked on? And this was a very large figure, and I just cut out the middle. You don't need to see all of them. What you see if you IP UPF1 is that within this window of 10 codons in either direction, okay, that it's roughly smoothly, let's call it, distributed across all these codons, some a little more, some a little less, but nothing after the termination codons. Likewise, if you do the same experiment in a UPF2 deletion, same thing. But with the dominant negative DE572 allele, which lacks UPF1 ATPase activity, now we see a substantial amount of ribosomes downstream of normal termination codons. And so from this, and from other experiments we had done earlier, we postulate that UPF1 must have some role in ribosome release. Imagine, if you will, that a premature termination codon gives rise to a poorly dissociable termination complex that needs a push to get out of there. We'll talk more later. Okay. Here's an important experiment with what happens with regard to stability of NMD transcripts when you delete one or both of the UPF1 binding sites on DCP2. Okay? If you just delete UPF1, what you find is that you get a whopping stabilization of these NMD substrates. If you delete one or the other UPF1 binding sites, you get almost no effect, very, very modest, and certainly nothing resembling this. And then if you delete both of them, you get more mRNA stabilization, but again, nothing to the extent of a deletion of UPF1, okay? And so UPF1 must have a function in addition to promoting mRNA decapping, and that's why I showed you that ribosome coverage of the termination codon regions. Okay. Now, what is it about a PTC that makes it attractive to NMD and not a normal termination codon? And so long ago we did toe printing. That's your classical toe printing experiment, primary extension inhibition, and showed that with premature termination codons, you get a 12 nucleotide toe print downstream of a PTC. If you look at normal termination codons, you never see such a toe print unless you do the experiment with a TSERF1, then you can see it. And so to a first approximation, premature termination is slow or inefficient. And here's another experiment that says premature termination is inefficient. Chan Wu in the lab put nonsense codons at all of these positions in the TPI1 mRNA and tagged the transcripts with HA at the 5' end and flag strep tag at the C terminus. And what she saw was that you could not detect premature termination products early on, and then they slowly increased, and only when the termination codon was near the end of the transcript did she see comparably abundant termination products. So here, quantitatively. So that suggested that termination efficiency increases across the coding region. And that was done by read-through as well in the luciferase, firefly luciferase gene, where she did essentially the same experiment, except now looked for the SF tag products, and read-through diminishes across the coding region, again, because termination's getting more and more efficient. Why is that all happening? Our best guess is that poly-A binding protein is influencing termination, okay? And that the more distant the translation event is from the end of the mRNA, the more modest the PAB effect is. Okay. So this got us thinking about termination a long time ago when we first started working on NMD. And we had a hint that premature termination was inefficient, and wondered whether now there might be an enhanced opportunity for near cognate tRNAs to compete with the release factors and generate a full-length protein. And we thought about it from the disease perspective, much as you heard this morning from Carrie Thrasher. There are a lot of diseases that are caused by nonsense mutations. And thank you, Thrasher. We both, Carrie, we both have the same number, 11% of all inherited diseases. And that means that on the planet, there are a lot of patients. So Stu Peltz and I, a couple of decades ago, decided that this was an opportunity to develop a read-through promoting drug that restores gene function for multiple diseases, and we started a company we called PTC Therapeutics. And I should add that this was after Dave Bedwell had shown in a really interesting pioneering paper that aminoglycosides would promote read-through of premature termination codons. Okay, so we started a company, hired a lot of people, and then over the course of about 5 years used MDX mice, that's the mouse equivalent of Duchenne muscular dystrophy, and CF mice to first screen in cell lines, then in animals, and slowly but surely develop a drug. We started with a screen of 800,000 compounds, found 7 interesting scaffolds, and ultimately had a development candidate that became the drug that was called atalurin. Atalurin is orally bioavailable. It's given to patients in a little sachet. They add it to the drink of their choice. And so no funky business about injections or anything like that. So it's orally bioavailable. It does not work on normal termination codons. It leads to insertion of the appropriate near cognate tRNAs at different nonsense codons, and you get full-length functional proteins that do everything they're supposed to do. It's well tolerated, does not affect NMD or mRNA levels, and it's not an antibiotic. Okay? And we've, for about 10 years now, been treating Duchenne muscular dystrophy patients with this drug. There are at least 1,000 patients on the drug worldwide. And there are a couple of major functions early on in Duchenne muscular dystrophy, and they are loss of ambulation and loss of pulmonary function. And this is what's called the STRIDE registry, where 200 plus patients have been monitored over many years and compared with regard to their ability to walk or to have FVC, force vital capacity. That's a lung function. Okay? And what you see is that patients on drug are doing better than patients without drug over those periods. So it's an interesting drug. However, it hasn't worked, hasn't succeeding in phase 3 for cystic fibrosis. Okay, so what's that all about? You know, do you just simply have a situation where you've got a nonsense codon, you give a patient a drug, and bingo, you get the protein? No, it's not that simple. First of all, you don't get that much read-through with Adalurin. You get maybe at most 10%, sometimes less. And does this work for any patient? We don't know. All of our patients that go into our trials have various genotypes, meaning nonsense mutation at this position, this position, this position, this position. They're not identical. And DMD is an X-linked disease, so you only have to worry about one transcript, but in CF you've got two. So it really can get complicated. And so we decided that what we need to know is more about termination, something that beyond just the nonsense codon. Everybody's known for a long time that UGA is the most suppressible nonsense codon, and that position 4 matters a lot. But we wanted to know what else mattered, and specifically, what about all the sequences around the termination codon? What about sequences downstream? What about the 3' UTR length? In other words, how influential is poly A binding protein? Okay. And so to address this, we started to do it first in yeast, where we did ribosome profiling and looked at read-through into what we call the extension region, which is the region from the first normal stop to the next stop, okay? And where read-through efficiency is defined as read-through reads per length in the extension over the coding region. We did this with a wild-type strain with a temperature-sensitive ERF1, that's what sub-45 is, okay? And then with data that we obtained from the web for sub-45 degron and RLA1 degron and HCR1 degron. That's data from Rachel and Nick and Allan Jacobson, okay? And we used the random forest analytical procedure to ask what were the influences of different sequences flanking nonsense codons relative to read-through. Okay, and this is the initial sort of data you do see in this extension region. You see a lot of read-through from the ERF1 TS. You see some from the RLA1 TS. You don't see it from any of the wild-type things. And so after much quantitation, this is what it looks like. Red is high read-through, blue is low read-through, and if you look at the stop, okay, what you see is that read-through efficiency is very low with UAA and highest with UGA, so everything's kosher with respect to what we know. Okay? And then there are degrees of influence of all these different nucleotides at all these different positions. Okay? This is a summary of what we saw. And then we went on and stole some more data from Rachel and did the same experiment with her HEC293 data. And remarkably, almost everything is the same. And for both, read-through efficiency increases with long UTRs. Okay? And 2 minutes. And this led us to say, okay, can we use these results to predict whether or not read-through will occur? So we have all this data on mRNA features, nucleotides in different positions, all derived from ribosome profiling experiments and analyzed using random forest. And then we got together with Dave Bedwell's lab and asked them to take the 15 most prominent CFTR nonsense alleles and do their dual luciferase assays and see what they get relative to what we would predict. And there is a God. The prediction relative to what they saw was quite good. And this is color-coded, so red are UAA mutations, yellow UAG, and green UGA. So untreated, these are G4-NA treated. Untreated, you don't have enough data because there's not that much read-through, so it's messy. But G4-NA treated showed that our predictability was good. And so this, I think, will allow us and others to find a way to sort the patients that are likely to respond from those that will not. And that's really important. Okay, so work in the lab in the last 5 years or so was largely done by Fung, Robin, Kocha, and Chan. The last experiments I showed you couldn't have been done without Dave and his team, especially Carrie. And the development of Etalaurin was done at PTC by a lot of people. And since this is a disease and therapy session, I want to highlight this last thing in yellow. If you are so lucky as to get something to the point of treating patients in clinical trials, remember, this is very difficult for them, and be understanding about that. They need your attention, your care, and they're looking for hope. So that's my closing message. As Dave Houseman used to say, anybody want to fight?
I will start with a question. Your two hybrid analysis and the structural models show clearly that UPF1 and 2 and UPF1 and DCP2 are not interacting at the same time. So how do you think about the handoff between those two states in the NMD pathway?
I think that when UPF2 and 3 exert their regulatory effect on UPF1, that's the beginning and end of their association, and that they dissociate having done that.
Thank you.
Allan, so you were showing when you deleted the two little segments from the DCP2 tail that bind UPF1, it wasn't as stabilizing as deleting UPF1 itself. And I'm just wondering, is it possible that UPF1 can be recruited to the DCP2 tail by interaction with one of the other decapping activators that's binding there?
We don't have interactions between the other decapping activators and the UPFs.
Through 2 hybrid?
Yeah.
But maybe there is, and just haven't detected it with 2 hybrid.
Okay. It's the old, well then, okay. You know.
Hi. In regards to the model in the end with the random forest, how do you think that might improve the therapeutic efficacy of the drug?
Random forest is a decision tree algorithm. And so when you have a lot of data and you say, as you work your way through this an enormous amount of, do you see read-through from this message at this position? Yes. What about this position with another message? What about this position with another message? The random forest algorithm lets you work through that yes, no decision tree. Thank you, with the aid of a computer. And so would it help you with your patients? No. You want the machine learning algorithm that we ended up with at the end, which you can download. That's what we used for prediction. We took all of the sequences we thought that were important and used them to predict which alleles were likely to be the most read through.
So, thank you for your talk. I wonder, the
So-
read-through compound
Ah.
and also currently the engineered tRNA therapy, they both show in ribosome profiling there's almost none of the native stop codon have been read through. I wonder, are they following the similar principles or mechanism that allows this kind of compound or engineered tRNA to only recognize premature termination codon, but not the native ones? Thank you.
So the thought we've had is that normal termination is highly efficient, perhaps by proximity to poly-A binding protein, perhaps by proximity to other regulatory factors on the 3' UTR. And so that high efficiency probably negates the ability of in this case, an ERF1 twinking drug to have any effect.
Yeah. Hi, Alan. Over here.
Matt, yeah.
Yeah, so in Neurospora, which is neither Saccharomyces nor mammal, we have the surveillance NMD, like you described, where URF will trigger NMD. And we also have 3' UTR introns, such as in Y14 and ERF1, and in EIF4A3, and two of those are part of the EJC. And there are CBP20 and CBP80, which are like the pre-cap binders. Those are critical for the EJC-mediated NMD through the 3' UTR intron, but not for the surveillance NMD. So there still yet may be a reason for that quote-unquote pioneer round.
Yeah, I was being a smart ass, you know that.
Hello?
Yes.
So I was wondering, now that you know more what is important for read-through, did you look back at the patients that responded well or not to etalurin?
You raised my least favorite point. So once we started paying attention to all this, I went back to my colleagues at PTC Therapeutics and I said, I need to see the genotypes for all the patients that were in the DMD trials and in the CF trials. And they said, sorry, you can't have it. And I said, why? And the answer is for every patient who enters into a clinical trial, there's a formal document that's an agreement about what you're going to do to that patient and with their data. Nobody anticipated this possibility, so there are no clauses in those agreements. And so the only way that I could get permission to see that data is to go back to all the patients' families and ask them to redo these agreements. And so far, that hasn't been worked out. That's the polite way of saying that. But that would be fabulous. And I'm going to a cystic fibrosis meeting in a couple of weeks to see if I might be able to liberate data from trials that were done on CF with aminoglycosides long ago. So it's possible. But not a, you just don't expect bureaucracy to get in the way of science. But that's what you have. Yes.
Hi, thank you very much. I was a bit curious about what you mentioned with regard to the position of the PTC and how efficient you can get the read-through, right? With the idea that when you were going closer to the end of the CDS, you would get better read-through, right? And that you thought it was...
Well, the end of the coding region gives you worse read-through.
Ah, okay.
So the curve is down.
Yeah, so then, because
It's consistent.
with the length of the 3' UTR. Yeah, thanks.
All right.
Thank you.
Join me in thanking Allan once again.